In this session, we're going to focus on one broad set of issues that Janet Woodcock brought up in her remarks this morning as being critical, and that's the right dose for the right patients, some of the challenges and opportunities in dose optimization. This is not something that should be viewed as simply as just setting two different doses and doing a pairwise comparison, as you heard, but given the heterogeneity in patients and some of the other issues that have been discussed today, is a potentially both more complex and more critical topic for the effective development and use of medication. So in this session, we're going to explore the importance of proper dose finding and dose exposure response characterization for successful drug development, approval, labeling, and life cycle management of products in patients who may need more individualized treatment than they've received in the past. We're going to start with a lead-off presentation from Vikram Sinha, the Director of the Division of Pharmacometrics of the Office of Clinical Pharmacology at Cedar. We're then going to explore dose optimization in more depth with case studies from Amita Joshi, Senior Director of Clinical Pharmacology at Genentech, and Brenda Cirancione, the Group Director of Clinical Pharmacology and Pharmacometrics at Bristol Myers Squibb. And then we're going to hear some additional perspectives from Sandy Allerheiligen, the Vice President of Pharmacokinetics and Pharmacometrics, Drug Metabolism, Quantitative Pharmacology, in Quantitative Pharmacology and Pharmacometrics at Merck, and Lissa Lavange, the Director of the Office of Biostatistics at Cedar, and Michael Maitland, Assistant Professor of Medicine at the University of Chicago School of Medicine. So Vikram, if you want to lead off for us, thank you. And once again, Ahimsa will be right here in front with the time cards.
Well, good morning, everybody, and it's really a pleasure to talk to you this morning about the challenge of dose selection in clinical development. So my charge is to set the stage probably for the next two speakers who will get into very specific examples in their respective areas. So I think the goals of the session for this particular session is, I think, to perhaps emphasize or re-emphasize the importance of dose finding and characterization of what I would call the exposure-response relationship, and the implications that this has in drug approval, labeling, and life cycle management. But I'm going to start on a different note and perhaps start with some of the challenges we see and with some of the actions. And in the course of the overall talk, give you a little bit more perspective on what's the existing framework for assessing dose response and current trends in approval, labeling, and life cycle management. So it's actually a little intimidating because Dr. Temple is here in the first row, and he actually wrote ICH-E4 to a large extent, and also the evidence of effectiveness. And I just wanted to start by saying that if any of you have not read these two documents, it's probably a good read. Many of these documents were written, some of them were written a decade or so ago, but the general principles in each of these documents still holds true. And to a large extent, it actually did lay out a very good path for actually characterizing dose response. But having said that, I think there are some challenges. And one of the big challenges is that actually the identification of an optimal dose is clearly not a requirement by law. It's desired. We want it for the patient. Prescribers would like to have more information in how to individualize treatment for various patients, but it's not a requirement. And not being a requirement can be good, can be bad, but clearly it's a balance that most development programs undertake in terms of development costs and cycle times. And by this, it means that there is often a challenge of how many doses to actually take to test in a trial, 2, 3, 4. There are cost implications, there are time implications, and clearly the charge for every drug development program is to try and get to phase 3 as quickly as possible. The other challenge, I think, is that the topic of dose selection is very different. I think we can conduct reasonably good dose-response studies. However, dose selection and the criteria that is the basis for it actually is very disease-specific. And the benefit-risk for a particular indication, to a large extent, often drives the types of doses or the number of doses that are actually selected. And the final topic is that even though, with great progress now over many years, with characterizing the exposure-response aspects, use of PKPD models, pharmacometric analyses, we still are perhaps at a point where these analyses are still designated as exploratory analysis. We would like to actually see this tag go away and take some form of being as good as any confirmatory analysis that can actually be used in the analysis of either phase 2 or phase 3 data. So very briefly, I think why invest in dose response? The first reason is phase 3 trials are very expensive, and you've got to get at least the brackets right in these particular trials. The identification of the right dose is, and I think should be, the key goal of every clinical development program. And this is for the obvious reasons. Too high a dose will cause toxicity, too low a dose, and you may get suboptimal efficacy. Now, moving a little bit back to sort of the general theme of the day, we are obviously trying to do this better in early development. And in early development, there are 2 or 3 key aspects that are important. One is the proof of concept. We obviously, as drug developers, you're interested in having concrete evidence that you've got a robust treatment effect. Those selections in terms of which dose to take forward into phase 3. And there's always this discussion of what is the minimum effective dose and what is the maximum safe dose. Now, the analysis of this, as at least stipulated in the guidance, can happen in numerous ways, one of which is pairwise comparison, but also documenting change in slope with changes in concentration, which is essentially an exposure-response analysis. And all this is very important because it helps one set the stage for how regulators are going to decide on the appropriateness of the right dose. Now, briefly to touch upon some guidance. There are 2 cornerstone guidance which actually help in shaping how dose response can be characterized. First is ICH-E4. This actually links dose response to safe and effective use of drugs, written in 1994 or published in 1994. It actually does a very good job of actually detailing the kinds of studies one would actually undertake, either a parallel design studies or crossover designs or titration studies, each of which has their merits, and how one can actually make an assessment of dose response. It actually, at least in my opinion, doesn't tell us much about how to select the dose. And it doesn't actually get into that aspect because it tends to be very subjective and disease-specific. The second guidance is the exposure-response guidance. And this guidance was a follow-on guidance to ICH-E4, which looked to link the role of concentrations with response. And it was geared, I think, to identify sort of the sources of variability that contribute to drug effect. So if actually one tries to look at this, the whole idea of using concentrations and relating it to drug response is to get a handle on the variability we see in the population. And I think Dr. Woodcock actually spoke about this in the morning, that when one limits a particular population and the focus on a particular population in phase 3, we are losing a lot of information about variability. Concentration is one way of getting at it and trying to make decisions about subpopulations and how to dose them. There are other guidances at the disease level which also speak to the specific assessment of dose response. And later on in the afternoon, I think you'll also hear a little bit more about the effectiveness guidance and all this, how all this feeds into the clinical evidence of effectiveness. Now, I think in all these guidance, there's actually a very common theme that conducting a dose response or a well-conducted dose response can and is primary supportive evidence of efficacy. And I think that's been written in many guidances routinely. I'm just going to give you a few trends of numbers we've accumulated over the years from the division and in the office. This is 2 slides talking to you about kinds of the number of doses that are typically studied in either a phase 2 or phase 3 setting. So here we've got a series of different therapeutic areas, the typical number of doses studied in phase 2, and the typical number of doses studied in phase 3. The 4th column speaks to the sort of the post-marketing commitments around dose response. So at least here we have 2 areas that have occasional PMRs or PMC that require further evaluation of dose response. And typically, as you'll see, that the number of strengths that are approved are typically around 1. Very few areas have more than 1 strength being actually approved for a particular indication. 3 of these areas, cardiorenal, psychiatry, and metabolism, actually have had a derived dose in the final label. By this, I mean that it's not necessarily a dose that's been actually studied empirically, but it's a derivative of our understanding of the dose-response relationship. A second part is looking at a few additional areas. And actually, the one area that actually clearly stands out is oncology. And clearly, those of you working in the area know the challenges that every oncology program faces in kind of conducting dose-response studies. We also had a recent workshop in May, which was co-sponsored by AACR, where this topic was actually discussed at great length. So what does this actually tell us? That we've got a spectrum of diseases, acute, chronic. The benefit-risk assessment across these different indications are going to be varied and largely sort of being weighed by unmet medical need. So it is actually very, very difficult to come up with any sort of concrete guidance that say a certain number of doses, this kind of a trial. And I think what we should be striving towards is more efficient and informative trial designs tailored for specific therapeutic areas. So I think each therapeutic area is going to have some unique aspects that drive the way these dose-response studies are going to be conducted. I'm going to run through very quickly some of the implications of dose in some of the recent approvals. The first one was for parathyroid hormone, and here a systems pharmacology approach was actually used to recommend an alternate dosing regimen. I'm sure Dr. Graf will be very pleased with that. In the derm area, we had exposure response analyses suggesting a need for a higher dose in subjects with higher body weight. And this was an analysis that was done after the phase 3 study was conducted. One in the area of oncology, and more recently, which probably gathered a lot of attention, was Apixaban for stroke reduction, where the exposure response analyses suggested a need for a dose adjustment in patients with normal renal function. So these actually give you a smattering of the different areas, and the dose still remains a critical topic for drug approval. We've also had some additional optimization aspects happen with certain indications. Here are 2 indications, both rare diseases, Cushing's disease and Gaucher's syndrome, where either a lower starting dose was approved based on interpolation of the exposure-response relationship for efficacy and safety. And in the case of eligustat, the entire program was actually conducted in a fixed dose setting, but a fixed dose was actually approved while all the studies were actually conducted in a titration-based design. So there was actually a derivation of a dosing regimen based on existing phase 3 trials. And in the case of nalexegol, the exposure response analyses was actually used to support a lower dose who could not actually tolerate a higher dose. So these kind of give you a flavor of how the importance of sort of dose selection in the final approval process. Very quickly, in terms of actions, I think from an agency's perspective, we expect to see very good dose selection, a rationale for dose selection as prior to starting phase 3. Dose-finding trials, we would like to either have a wide range of doses studied and or to use model-based approaches. By that I mean using concentration response analysis to understand the shape of the dose-response curve. Many therapeutic areas are targeting the minimum dose with near maximum efficacy. So I think the inflection points of these curves are becoming more and more important. And clearly the move is towards rational dose selection. So you can actually see from the table that some areas do a lot more in phase 2 and some areas do a lot less in phase 2. And clearly, I think moving forward, we expect to see more efficient and innovative trial designs and analyses approaches that look to integrate all the information. I'm going to actually end with maybe this table here, which actually we've used on multiple occasions, and trying to sort of paint a picture for why this is sort of at the center of what clinical pharmacology does. And you can actually, for those of you who are involved in the drug development process, your early trials are trying to get at some aspect of MTD. And so we would like to see sort of good dose ranging studies, the use of modeling and simulation to design trials, looking at ways to integrate the information from these trials to give us information about alternate dosing regimens. And all this information should be happening more readily during the IND stage. We don't see a great deal of covariate-based dosing in registration trials, so this is something we would like to actually see more of. And finally, I think there's very few NDAs and BLAs right now who don't have a significant component of exposure-response analyses. So I think this is a trend, and I think we look forward to actually having these analyses and helping us understand and help us in the dose selection process. Thank you very much.
Thanks, Vikram. Amita?
Okay. All right. Good afternoon. All right. I want to acknowledge my co-authors here on this presentation, my colleagues at Genentech. So I want to just open up with telling you a little bit about some of the literature, some of the review we have done with oncology drugs approved in the last few years. So we looked at the oncology drugs approved, the first approval of oncology drugs in the US. And of the 41 drugs that were approved, 11 had post-marketing commitments for dose optimization, which were either studying higher or lower doses or further data analysis of existing trials. So 27% of drugs in oncology had some additional work that was needed. Now, if you overlay on this, in the blue boxes, the drugs that were developed at the maximum tolerated dose, you see that 50% of the drugs were actually developed at the maximum tolerated dose. And of that group of molecules that were developed at the maximum tolerated dose, 43% had dose optimization PMR, PMC. So clearly we need to take a different approach, not develop drugs at MTD, and actually have a different way of identifying doses. And that's what I'm going to focus on. There's clearly understanding in the oncology space now in terms of having better dose optimization in phase 2 trials. So I'm going to tell you a little bit about some of the approaches I believe will work. So overall, I believe there are two big approaches. The first approach is design, study design approaches. So how could we improve productivity and success in oncology? The first approach is by designing trials in different way, having multiple doses to study, understanding the dose response better, having adaptive designs, making sure we have really good ways of making decisions during a trial so that we can keep the doses that work and discontinue doses that don't. Another important thing is collecting tumor biopsies, because in oncology, tumor-based biopsies and tumor-based biomarkers are very important. Because unless you get them, you really don't have a good way of assessing site of action effect. So getting those and having good assessment of tumor biopsies, having good methodology set up, having good collaborators in the clinic who are committed to doing it is very important. Next, surrogate endpoints. As they're getting developed, more and more surrogate endpoints are being developed in oncology. Using them, for example, minimum residual disease in heme malignancies or pathological complete response. It takes a lot of work to develop these endpoints, but companies are doing that, and the regulators are very open to that. Another one, this design aspect is really understanding the relationship between early and late response, early and late endpoints, so that we can actually apply some of these early endpoints in phase 2 trials. So we are better, more successful in the later trials with the late endpoints. So those are some of the study design aspects. The second aspect that I want to talk about are better prediction tools. And we've talked about that in the earlier panels as well. We need better prediction tools for PK, PD, safety, and efficacy, so that we can actually understand this therapeutic window. And in oncology, generally, the window is very narrow. So how do we really thread the needle? And you can use lots of different predictive tools to better inform decisions. And I'm going to tell you a little bit about all these, give some examples in these areas. So all the tick bars here are examples that I'm going to show you, one slide per case study, and then on the bottom here sort of indicates where I'm talking about efficacy or safety in that particular example, just to make it simple. The first example is an adaptive trial design that we've recently adopted. So in this case, we were studying TDM1 Kadcyla in gastric cancer, and Kadcyla is approved in metastatic breast cancer, HER2-positive metastatic breast cancer, and we were studying it in HER2-positive gastric cancer. We knew from trastuzumab phase 3 trials that there is a dose-response relationship in that patients who have lower exposure are not getting benefit from the drug. And actually, we have a PMR study for trastuzumab to address this. So we wanted to be clever here in developing TDM1, which is trastuzumab, which has a toxin attached to it. So we designed a phase 2-3 adaptive trial where we had 2 stages in this trial. The first stage where the standard dose, which is the 3.6 mg per kg every 3 weeks, and the second dose, 2.4 mg per kg weekly. Now we changed the dosing schedule here from 3 weeks dosing to weekly dosing, but in effect, we were able to double the dose. And so the stage 1 of this trial will read out and inform the stage 2, where 1 dose will be picked for the phase 3 portion of the trial. This is just a conceptual now example that shows various approaches that we have applied. Here we generally use xenograft, human tumor-derived xenografts in oncology to assess dose exposure response. In animals, translate that to humans, and then there's also early to late translation. So there are multiple approaches that could be applied in all of these areas. Here is an example of how we have used translational PKPD in using tumor dynamics. So it's a simple 2-stage approach where xenograft mouse data regarding exposure and efficacy is modeled to give an exposure response, and then human PK data is brought in, and then assuming that the response in humans is similar to the response in the animals, tumor growth inhibition model is developed. My colleagues at Genentech actually have done some retrospective analysis of 8 anti-cancer compounds, and they've shown that the simulated tumor growth inhibition correlates with overall response in the clinics. These are 8 anti-cancer drugs that have been approved. And of course, you have to have relevant animal models. These are not low-bar animal models. These are the relevant animal models, and also relevant drug exposure has to be achieved. So what we have done is, using 60% as a cutoff. So early data from preclinical models helps us identify whether we are going to achieve a particular target concentration in the clinic or not. This also helps us with picking the right doses, doing dose escalations fast, and actually getting to the target concentration quickly in patients. Another example I want to show you is using clinical PKPD to pick doses. Here on the right panel is a drug, a PI3K molecule in the clinic being combined with standard of care, and PKPD modeling using tumor dynamics in the clinic has been developed for this combination. On the left here is a predicted clinical tumor response the very top curve, top line here is the predicted response with standard of care. And there are many lines here. The two green lines are the low and the high drug cohorts. And the team was trying to figure out whether you could change the dosing of the high dose in order to modulate toxicity, because in this case, toxicity was limiting dosing in these patients. So again, the same simulation paradigm was used to look at different dosing schedules, 21 days on, 7 days off, and so on. And the team had more confidence in changing the dosing schedule and assessing it because they had more confidence now that it would not really change the response as much as they were worried. I'm going to skip this one. I'll go to this example. So I mentioned tumor-based biomarkers. So here's an example where we are targeting the PI3K pathway and targeting AKT in particular with a small molecule. And the green circles are the biomarkers, the downstream biomarkers that are being measured. And these are downstream to target engagement, so they actually indicate some activity downstream. So on the right panel here, you see an exposure response curve showing the relationship between area under the curve at 3 different doses and the biomarker suppression. And this showed that at 400 milligram, you had 80% target inhibition, and so the team had more confidence in picking the dose for this. And at 200 milligram, you did not have as much target inhibition or didn't have complete target inhibition. Again, very important to have tumor biopsies and to use them appropriately. Here is an example where I want to show how in oncology we use a lot of single-arm trials. So there is no control arm to compare your trial, data from your trial. So how do you address it? We have used an approach here that is literature-based meta-analysis. So on the left here is a paclitaxel literature-based meta-analysis where we curated data, trial-level data from the literature, from 55 references, 49 trials, 95 arms, and so on, more than 4,000 patients. On the bottom here is the dose response of the administered dose versus neutropenia. The red circles are weekly dosing paclitaxel, and the blue circles are the every 3-week dosing paclitaxel. And the size of the circle is the number of patients. And you can see superimposed here in triangle are the test drug along with paclitaxel. So despite not having paclitaxel in the trial, by using this literature meta-analysis, we were able to benchmark our drug with paclitaxel and gave us better confidence in moving doses forward. We also, on the right side, did PKPD, traditional PKPD analysis to further validate that this was indeed the case, and both these were able to give us increased confidence, and we were able to move forward. I want to give a shout out to systems approaches that are also being applied in oncology now. So here again, in the middle, I show the PI3K and the RAS-RAF-MEK pathways that are targeted in oncology, multiple drugs being studied, and multiple drugs being combined also. And we are modeling not only the activity, but also toxicity. In this case, there are a lot of toxicities that are overlapping. And because of the feedback mechanisms and the overlapping systems information that we have in these models, we really have better confidence in what we are, when we are deciding to move forward with it. Knowing which brakes to put on and when is really important. So in summary, identifying the optimal dose is a primary challenge, and uncertainty. I showed you earlier that 30% of oncology drugs have some dose-related PMR, PMC activities. I showed you some approaches, adaptive trial designs, and actually using translational approaches and PKPD very early on, and actually increasing the number of tools you bring to the table, and just having fun doing it. I think we are actually making a lot of difference here in terms of bringing quantitative data to the table and really making an impact in changing patients' lives. Thank you.
Thanks, Amitar. Next cases will be presented by Brenda.
Okay, thank you very much. I'm going to present a case study on the dose selection for Apixaban, specifically with a focus on the atrial fibrillation indication. Disclosures. Okay, when you think about selecting a dose for a compound, we're all doing the same thing, and that's balancing between safety and efficacy of a product. So for the anticoagulation space, we're balancing between identifying optimal prevention of thrombosis and balancing that against finding an acceptable level of risk of bleeding. Apixaban is a factor Xa inhibitor. The factor Xa inhibitor enzyme was chosen because of its location in the anticoagulation pathway. In addition, inhibition of the factor Xa enzyme does not interfere with the activity of the residual thrombin activity in the formation of thrombosis. In addition to the target-selected enzyme, the clinical pharmacology profile was identified early on to have an ideal profile for our target population, which is an elderly population. So we want something that has a low metabolic distribution, small volume of distribution, and multiple elimination pathways. So something that's going to have limited sensitivity to differences in patients with renal function, something that's going to be tolerable to multiple drug-drug interactions. The clinical pharmacology profile from Apixaban was evaluated in several trials and has shown that it has achieved the target profile. It has a rapidly absorbed oral bioavailability of about 50%. It is not affected by food, pH, or therapeutic dose. The half-life is suitable for once-a-day or twice-a-day administration. It does indeed have multiple routes of elimination, with 27% eliminated renally, and it's also metabolized by cytochrome C450 enzymes. There's no active metabolites of Apixaban and has limited influence of demographics and disease characteristics. So overall, this product profile led us to a place where we thought this was great going forward and it would be suitable for our population. The challenge comes in when you're taking that anticoagulation and atrial fibrillation and running a phase 2 trial. Traditionally, you would run your exposure response dose ranging study in phase 2. This would be a large study with AF. The event rate is so low that the size of the trial that would be required to identify this in the target population would be on the size of phase 3 design. Not suitable. So what was done was the phase 2 study that served to support the dose selection for multiple phase 3 programs in different indications was a daily dose selection for VTE, prevention, and total knee replacement. This was a 12-day study where patients were about 12,000 patients were enrolled, 1,200, sorry, and we could evaluate ranges of 5, 10, and 20 of Apixaban in 2 comparator arms. The Apixaban doses were further then divided into QD and BID regimen to allow us to compare once-a-day versus twice-a-day dosing in a dose-ranging study. What you're seeing on the slide here is the results of this phase 2 trial. On the right-hand side, you see the safety, total bleeding. And if you look at the 5 milligram total daily dose, the rates are less than that of the 2 comparator arms shown in gray, supporting that the 5 milligram dose, total daily dose, actually results in a better safety profile than the comparator arms. And as expected, as you increase the dose, you'll see the rising event rate above the comparator arm up to the 20 milligram dose. And this led the team to conclude that the 5 milligram dose from a safety perspective was the right total daily dose to take forward. Balancing that for efficacy, if you look on the left-hand side, you see the VTE rates. And you'll notice that the Apixaban arms performed better or superior to the comparator arms across all dose ranges. So further supporting, we're going to take 5 when you balance that against the safety profile. But then the question comes in of QD versus BID dosing regimens. You'll notice on the efficacy side, the orange bars represent the BID dosing regimens, and the purple bars represent the once-a-day dosing. In all cases at all dose levels, the BID arm performs better efficaciously than the QD arms. But the choice to take BID versus QD is always a challenging one, because now you're balancing convenience of therapy versus response and safety. So we looked at this in several ways. This is an illustration of a simulation of the PK profile for the 5 milligram total daily dose given once a day and twice a day. And what you'll notice here is that the twice-a-day dose results in a lower peak concentration and higher troughs, consistent with a more stable profile for anticoagulation. Lower peaks are likely to give us a reduced bleeding risk, And the higher troughs are going to provide us a more stable efficacy throughout the profile. Another way that was looked at to analyze this data from this trial was to combine the efficacy and safety into a single endpoint. This is called our Therapeutic Utility Index. A score of 100% on your Therapeutic Utility Index, as a reference, is a perfect drug. So 100% efficacy and absolutely no safety, just to give everybody an orient where they are. So what you're looking at on this display is on the vertical axis, you have the Therapeutic Utility Index, and across the bottom you have the overall drug exposure, expressed as total daily dose, total daily AUC. You'll see two lines. There's a purple line and an orange dashed line for twice daily and once daily dosing, and the Therapeutic Utility Index across the range of exposures. You'll notice they both do decline as we increase exposure, and that's reflective of increased safety contribution as we become higher in exposures. Of interest, when you look down on the lower end of the exposure and across actually all range, the twice daily dose has a higher Therapeutic Utility Index than the once daily dose, further supporting the use of a BID regimen over a once daily regimen. On the lowest end of the exposure, you'll notice that the twice daily dose actually has a higher Therapeutic Utility Index than both enoxaparin and warfarin, so higher than both the comparator arms. And the exposures in that region, on the bottom you'll notice the box plots, are associated also with the 5 milligram total daily dose. So based on all of these analyses, for VTE prevention, a 5 milligram twice daily dose was taken forward. But this isn't for VTE prevention, this is for AF. The team concluded that a higher exposure would be required for AF indication. So it was up to 10 milligrams daily dose. Then, consistent with the results of the QD versus BID trial, they decided to use a twice daily regimen in their phase 3 AF program. The primary dose taken into phase 3 was the 5 milligram BID dose. But it is worth acknowledging that not all patients are at the same risk for bleeding, regardless of what drug you take. There's a subpopulation of people, elderly, lightweight females, who are more likely, for example, to have increased bleeding. So a sub-algorithm was devised for them. Within the Apixaban program, demographic characteristics and contributions of things such as age, body weight, and gender were assessed in both phase 1 trials as well as in population PK analyses. While none of these factors on their own contributed a significant enough an increase in exposure or in risk of safety to warrant a dose adjustment, it was acknowledged that the combination of more than one of these would actually lead to an increased exposure in a subject. Couple that with the fact that many of these characteristics also underline an increased probability of bleeding. An algorithm was devised such that patients who have at least 2 of the following criteria, age greater than 80, body weight less than 60, serum creatinine greater than 1.5, would receive the 2.5 milligram dose in the phase 3 program. The results, this design was evaluated in 2 phase 3 programs. The ARISTOTLE study evaluated Apixaban versus warfarin, and the second trial, the AVERROES study, evaluated Apixaban versus aspirin for those subjects that could not tolerate vitamin K antagonist therapy. The results stand for themselves. The ARISTOTLE program showed superiority across all the endpoints, safety and efficacy, with the target against warfarin that was monitored with the designed dosing regimen. Similarly, the AVERROES study also demonstrated superiority in terms of efficacy with no significant differences in bleeding. So overall, the Apixaban program was a rationally designed factor Xa inhibitor. The clinical pharmacology program was discussed and thought out early into the program as to what the target profile they wanted to achieve was. The twice-daily dosing was based on a clear rationale based on multiple analyses, as well as the appearance and the observation from the data of a more stable anticoagulant profile. This choice for the twice-daily dose reflects the greater priority placed on having a decreased variability in our anticoagulation results and clinical outcomes than on the convenience of the once-a-day dosing regimen. The BID regimen and doses selected have demonstrated favorable outcomes in multiple phase 3 programs across different indications. So overall, the twice-daily dosing delivered beneficial therapeutic balance between efficacy and safety across multiple indications with the dosings that were evaluated during the phase 2 and 3 programs. I do have to acknowledge this work is a contribution of a large group of people who have worked extremely hard in the BMS-Pfizer Alliance team. The names on this slide just represent a small portion of people who have contributed to the works shown on these slides. There is a large group of people that have contributed greatly. Thank you.
Brenda, thanks very much. Next we'll hear a perspective from Sandy.
Hi. Well, since I'm kicking off the panel, I wanted to just talk with you and, how are we going to go forward? So some of the notes that, as I was thinking about this over the weeks and especially today, I think Dr. Woodcock gave us a great kickoff. And the challenge that she described, and the other speakers as well, it's, we're challenged to reduce the cost. We've seen, it's like, what's our problem? We haven't been able to get out new drugs to patients. We've dramatically increased the cost. And she brought it down to two development styles. One is check the box. We do this at this stage. It's a very phase appropriate, staged, and it's run to phase 3. The other is spend more time and actually learn. And I had to laugh. I thought, boy, this is back to the future. I remember these conversations 25 years ago. And so what makes it different? One of the big differences now is, one, we've actually got examples that may work. And we had some beautiful ones today. And then the other pieces that are really new are the what we know about biology, our ability to access it, the new in vitro and in vivo tools, as well as the quantitative tools. There were days a simple PKPD model took hours to run. That takes seconds. So what we now have many more possible things, and it really gives us a chance to get the dose right. But I'm going to say this several times, and I think Vikram said it in the beginning, there's no checklist. Every dose is different. Every drug is different. And if we come in with the mindset, well, this worked last time, it's going to lead us down a path. So we have to use all the tools, and everything should be about the patient. And if I could suggest anything, it's about seamless discovery and development. It's not about separating, okay, we're going to do discovery now, and we're going to do translational PKPD, and now we're going to do phase 1, and now we're going to go to patients. It's really coming back to what are the questions and what are the tools we need to solve them. It's not about rewarding folks for the most complex models. That doesn't matter. It's about, did it answer the question? Were we able to communicate it? So I would propose that the early learning is our glue. It's our chance to experiment, to really understand, get into patients as quickly as we can. Why do the single ascending dose and multiple ascending dose studies? Why are those so often in healthy volunteers? Can we challenge that learning? And Vikram challenged us this morning about it's much more about understanding the variability. And to me, I will come back to that, is it's really understanding the real-world patient. So you're going to hear me say this again more than once as well, and it was coined by one of our scientists, that, we spent so much time thinking about translating the discovery, learning, and in the animal models to the clinic. Perhaps the more relative translation is translating the clinical trial patient to the real-world patient. And how do we do that? Because so often, if dose is wrong, it's because we've been focusing on a special clinical trial patient and not the real-world patient. Brenda did a lovely job talking about how we're going to get to that clinical trial patient. So it can't be about the tools. It really has to be about that cyclical learning. So our process is not linear. We've got to get away. It's next to impossible to put it on a slide, but most of us think of it linearly, and we need to take that every time we run an experiment, whether it's non-clinical or clinical, we need to take that learning bidirectional. Does this inform the patient? Does this change our way of thinking? Pete said, well, what if we get the models wrong? We already know that. We only get the model. The models are wrong. We have they are a repository of our understanding of the data to date. If they're wrong, when the data come in, that can be the greatest tool to see a big breakthrough. But it's about bringing it all back together. But let me do one quick commercial. If we have, I don't think you can ever validate a model. I think you qualify it to help inform. And it is only as good as the data and knowledge we have. But it does allow us to test what if we're wrong and to truly explore and do parameter sensitivity. So my time is up, so I'd like to summarize in many ways what I think we've described is quantitative and systems pharmacology. And I would encourage us, as we think of these 2 morning sessions, put the "and" back in, quantitative and systems pharmacology. It's bringing the 2 pieces together. They're not separate. Again, qualify your model, understand your data. That's our if we want to quit being just an aside and exploratory, then we have to be as serious about that and be able to communicate it. It's about training. The greatest folks that impact people can have is those that can scope and understand the problem and figure out the tool you need to answer that question and get the dose right. It's not about PKPD, it's not about PBBK, it's not about systems pharmacology. It's about integrating and understanding when to use what tool, and so training our folks so that they can do that. Thank you. I look forward to the discussion.
And next is Lissa.
Okay, I'm glad to not be the first person without slides. But I have a feeling I might be the token statistician here. Are there any other statisticians in the audience? Maybe just a couple? No? Okay. All right. Well, I'm happy to take any and all questions. So thank you for including me, Vikram, in the panel and the organizers, Mark, and the folks at Brookings. I'll have a few comments or remarks that bring a statistical perspective or add a statistical perspective to the discussion so far. And my remarks will, in part, reflect my position at FDA. I oversee about 180 statistical reviewers in CDER. We're a big and important group. But my remarks are undoubtedly personalized a little bit by my experience with clinical trials in both academia and in industry prior to my coming to FDA. So as a statistician in CDER, we absolutely recognize the importance of finding the right dose as early as possible and the risk of failure in later phases, or phase 3 in particular, if we don't find the right dose. And, you know, dose finding is not always viewed as our domain or our purview. And in fact, some phase 2 protocols might go through CDER without a full statistical or detailed statistical review. But in areas like oncology, of course, we pay a lot of attention to dose finding. And in more and more areas, we're trying to do this as well. When we do get a full statistical review of a dose finding study, we should and are, as far as I know, very accepting of model-based methods because we recognize, obviously, the efficiencies. So methods like continual reassessment methods or other response adaptive randomization methods that make efficient uses of patients and other resources are very important. So if this isn't our domain, then what is it that maybe we can add to the discussion, or what good can a statistical perspective do here? And I think there's a couple of things, and because of this, we are getting a little more involved in this area. One is a healthy respect for uncertainty. Vikram already mentioned the importance of uncertainty, and he hears this from us a lot, I think. You know, dose-response relationships can look really pretty with PK and PD parameters, and then you get into the big bad world of clinical outcomes, measures of how patients feel and function and survive, of course. And you've got more variability in the assessment of these outcomes. You're also typically in a larger study when you're trying to see signals with respect to clinical outcomes. And that larger size, larger number of patients brings heterogeneity in the patient, in their response to the dose, and so forth. And so these pretty dose-response curves that you had been so comfortable with all of a sudden in the clinical setting don't look so pretty. And sometimes, you know, having a better ability to quantify the uncertainty can help at least interpret the results, or maybe figure out if it's a disappointment or if it's just expected. Also, it's often the case when you get to the later phase studies that we really aren't looking at the full dose range, right? It's very difficult to justify economically taking a lot of ineffective doses to phase 3. And so if you're only looking at a narrow range of the doses, you're in the upper part of the curve, then you don't see the dose-response relationship. Who's surprised? But, just having some reality checks about interpreting what you see with the clinical studies, particularly later on in development, I think is something the statistician can bring to the table. The other reason this comes into our domain is that more and more, as Dr. Woodcock spoke this morning, we are seeing programs where we have a sturdy phase 3 study and we need additional evidence from clinical pharmacology or from phase 2, and this is described in the clinical evidence guidance. So phase 2 studies that might have been designed to find the right dose through an exploratory analysis all of a sudden are being asked to carry the weight of being a phase 3, to being used as confirmatory. And this is where I think some of the problems, or maybe some of the comments from my group, probably come in, hopefully not too startlingly. So what can we do? Well, traditionally, if you think about dose finding as a statistician, we're trained, very logically, dating back to some landmark papers from the early 90s in dose finding in clinical trials, that there's a sequence of questions, right? The first question is there a signal at all? Is the drug effective or has activity? And the second is, are there any doses that differ from control? And the third is, what is the dose-response relationship? And then the fourth is, what's the best dose? And how do you work through those sequences? And again, historically, statisticians would come in on that fourth question mostly, very powerfully, and want confirmation that we've got the right dose. And confirmation means we want the type 1 error to be controlled. We want to know what the chance of a false positive conclusion is. So we tended historically to favor methods that are based on multiple comparisons because we know how they behave. But we also recognize that that's not always the most efficient way to go because it doesn't use information across all the doses, and it certainly doesn't use any information interpolating. It just evaluates each dose separately. Now, that's maybe the conservative side of the spectrum, and the liberal side of the spectrum are the model-based methods that we are fully embracing in phase 2 and early dose finding, but are more nervous about in confirmatory settings. And something very exciting has been happening in the last few years. Some statisticians in industry have developed some methods that combine these two, and the statisticians in the regulatory agencies on both sides of the ocean are very excited about this. An example of a combination is the MCP mod procedure. Not to give a plug to a particular procedure, but many of you may have heard of it. But it's a multiple comparisons and modeling procedure that actually takes advantage of a powerful and efficient model, but also has built into it multiplicity control, so that when you not only find the model, you can use it to estimate the dose and have some good assurance that the dose you've estimated is optimal based on the model being the right fit. And this is nice because model-based methods by themselves, are very dependent on the model being correct, and you don't usually know if the model's correct when you start out. So having procedures that build in some multiple comparisons control into the model selection, can be an advantage. The EMA actually, I can't end just yet, but almost. The EMA qualified this method recently, and it's currently being reviewed by my office and the Office of Clinical Pharmacology, and we're anticipating an endorsement from our side as well. And why do we care? Because there's a lot of bad dose-finding stuff happening out there. So anything we can do to endorse a better and more powerful method that's efficient but also has good statistical properties, we feel is a contribution. So 2 ending thoughts. One thing I'll mention, and maybe it's provocative, maybe it's not, and I've talked internally to Dr. Temple and Dr. Zena Isam and others, but, how to personalize dose selection is of interest, and it's a little bit of a catch-22. We may need the data collection in a big phase 3 study to get enough range on the responses to have good exposure-response modeling and be able to figure out the dose. But you are in the confirmation stage, and where statisticians sometimes feel we get into trouble is when we try to take a relationship and use the same study to go back and confirm the subgroup that needs a dose adjustment or, some kind of modification. I mean, it's a pretty basic principle of science that using the same data to fit a model and then to validate the model is a bad idea, right? So, this, and all statisticians are suspicious of subgroups, right? Particularly post-hoc subgroups that you didn't specify in advance. That's just, we have a gene. I think it's a genetic tendency that we have. So, is there a day where we could get enough data to do the good exposure response modeling so that when we get to phase 3, we randomize patients to the dosing algorithm, and then the comparison is whether a patient's dosed the right way or better than patients dosed the wrong way. And that's something that I think could be really wonderful for the public health, but a lot of things would have to happen. And then finally, because I didn't see any more statisticians on the rest of the panels, I just want to make a plug. We are all about trying to innovate, and it's a big push in the Office of Biostatistics at CEDAR. We are doing a lot of work with predictive and prognostic biomarkers, surrogate outcomes. We're doing a lot of work with enrichment designs. Optimizing dose is one thing, finding the patients that really benefit from the drug is another. There's a lot of exciting statistical designs for clinical trials that look at different methods of enriching. We've had a lot of speakers come into the office recently to talk about some of these, and if there's an opportunity, I'll make some comments this afternoon about them when we get to those topics. But those are my remarks on dose finding. Thanks.
And next, Michael Maitland.
Thank you, Dr. McClellan, and to your team for inviting me to meet with everybody today. It's exciting and gratifying to see what colleagues in the field of clinical pharmacology are accomplishing in drug development. These are examples we need to remember of really best execution. As Sandy was alluding to, I think part of why we are here today is to brainstorm how we can expand these efforts, but it's important to bear in mind that most drug development efforts have less effective and much less thorough analysis. And part of that is because there is this limited pool of individuals who can conduct this work effectively. Any approaches to scaling up, as was mentioned earlier, are going to require some serious consideration of how we expand the workforce. I think that includes specialists who can lead these efforts and conduct these efforts, but also bear in mind my physician colleagues are, with all of their new advances, are squeezing the early curriculum, at least in American medical schools, to have less and less time devoted to pharmacology. And so there need to be some efforts to better educate the entire workforce in that space. In my clinical practice of oncology, we're facing perhaps some of the unintended consequences of this success. Breakthrough designation, I think, is very exciting and helpful, but has created a new sort of high-stakes race with the development of new compounds, and when there are more than one compound and more than one sponsor, inevitably a race. And that race is a complex one. I don't think anyone has had time yet to figure out how to win, because there's one strategy that says get to market as quickly as possible. There's another strategy that says let's understand at least a little bit more about our compound in order to have a better product, even if we are second or later to market. The experience of one of our patients at University of Chicago, I think, reflects the significance of the recent advances and the new challenges that we face. And I'm trying to advance the slides here, in effect. Okay. So the patient is KS. He's now 69. He barely ever smoked cigarettes in his life. When he was 66 in June of 2012, he unfortunately developed progressive shortness of breath and ultimately was found to have lung adenocarcinoma with a collection of fluid around his left lung, a pleural effusion, which meant that his disease was stage 4 and incurable. His tumor was found on relatively rudimentary molecular testing at the time to have a translocation of the EML4 gene, linking it to the ALK gene. From now on, I'll call this the ALK translocation, which meant instead of chemotherapy, he could be treated with the oral compound crizotinib Xalkori, developed and marketed by Pfizer. He did well with this treatment, so much so to the point that in December 2013, 18 months later, all clinical evaluations suggested no evidence of active disease at that time. By that time, there were 5 compounds in active development anticipating that his disease would become resistant to the frontline crizotinib and aiming to be the new second-line standard of care. 5 months later, the first of these was approved, ceritinib Zycadia, developed and marketed by Novartis. Leaders in oncology celebrated this event as a milestone, the equivalent of someone running a 3-minute mile. The urgency was appropriate. We had this new cohort of patients who would do well for a time with one drug, only then to have progression of their disease and ultimately die without any better treatment options than standard palliative chemotherapy. Time won't allow me to delve into the details here, but for this class of drugs in this setting, there were some real challenges in determining a tolerable and effective dose. By the time KS had his disease progress, there was clear evidence that the investigational agent, alectinib, had better tolerability based on dose and examination of food effects on drug exposure. He did not want to wait for the time it would take to get access to that compound through an expanded access protocol, and he decided with his clinician at the time of his disease progression to start ceritinib treatment. He started at an intermediate dose, not the labeled dose, out of concern he may not tolerate the drug well. And instantaneously, his symptoms of pain improved. That's a little bit of an exaggeration on my part. Within 3 days, though, he was doing much better and was so enthusiastic about this, he decided on his own to increase his dose to the maximum labeled dose. The consequence of that, as one might anticipate, was a few days later, he had developed severe nausea, vomiting, ultimately volume depletion that required IV fluids in the clinic, as well as a holiday from the treatment. That experience left him somewhat debilitated and took a prolonged period of recovery. He then starts back on the lower dose of ceritinib and then unfortunately has this same experience more severely and ends up hospitalized and again requires a holiday from treatment. At 69 years old, this experience left him so deconditioned. We're now many months later where fortunately his cancer is relatively at the same magnitude of burden as when he first started ceritinib, but it's still unclear whether he has the overall physiologic capacity to move on to treatment with another compound like alectinib. As the prior speakers highlighted, this is exactly the kind of patient-level experience that none of our clinical trials capture and is critical data that we are readily capable of collecting, but not incentivized or readily capable of sharing in 2015. And so I think this highlights the key point for our future. This is a commercial, but you know now that we are not experts at University of Chicago in delivering precision medicine, certainly not in this patient's case. But we and every other academic medical center in the country are promoting ourselves as being experts in this space. And so what it highlights, though, is a key point, is that success is breeding the new challenges. We get better at developing therapeutics for smaller populations. The next natural step is to get the treatment and dose right for ever smaller populations. And others have thought about paradigm-changing approaches to how we might develop drugs in this environment and expand the sphere for productivity in research and development. Sandy, I decided to one-up you and generate a slide that might help us to establish this concept. This is a beautiful article in Science Translational Medicine a few years ago, put together by a team of consultants and patient advocacy leaders to think about eliminating this concept of the linear drug development pipeline and instead thinking more about a network. They use this term NETs to catch new therapies, and the strained acronym is Navigating the Ecosystem of Translational Sciences. I won't run you through the diagram now, but as you can see, it's quite complex, but I think relatively reflective of what actually is happening in drug development today. And then finally, I just want to touch on one more point. It's raised earlier by Dr. Rog's quotation of William Gibson, is that at our leading academic medical centers, I think we are having a lot of experiences with patient care, use of investigational therapies and newly approved therapies, and the cutting edge of basic science, which is leading us to experience opportunities now that just haven't yet been distributed elsewhere. And what we have the opportunity to do in this forum, and hopefully through tomorrow, is anticipate this distribution and what are the challenges in terms of regulating that environment in order to get better outcomes for patients. And what's featured here from Brannon and Sawyers, again published a couple of years ago, is this concept of studying the individual patient, performing an N of 1 investigation. And that is the reduction ad absurdum for our efforts to get dose right first in populations and ultimately narrow it down to the individual patient. And my main point here is what's promoted in this paper, what's been mentioned by prior speakers, is the continuum of evaluating dose need not, and I would argue should not stop at the time a drug is approved for marketing, but rather should continue on through more effective collaborations among not just sponsors and regulators, but also health systems and perhaps patients themselves to get better results and for everyone to succeed in what's a very exciting new area of medicine and medical care. So with that, I don't want to get any further in people's way of lunch. Thank you for your time and attention.
Thanks very much. I'd like to, again, thank all of our presenters for some excellent presentations. You see, we are kind of at the lunch hour. I don't want to stand in the way of that for too long, but I would like to allow some brief time for a few questions or comments if there are any, and don't worry, we will leave you time for lunch after this session. So please go ahead.
Can you hear me okay?
I think you're on now.
Okay. So I want to talk a little bit about this use of phase 3 for dose optimization or exploration. We've seen, in general, 2 approaches and different levels of comfort with these approaches in the regulatory context. The first is trying to do something right by design, taking multiple doses into phase 3 or doing some proactive adjustment based on some intrinsic patient factor. The second approach is just study 1 dose, if we're lucky to collect some blood samples and then do this kind of post hoc exposure response. And I have to say, I'm being overly simplistic, but there's varying degrees of comfort amongst regulatory bodies and reviewers in different disciplines. It's not just, we won't pick on statistics, Alyssa. I guess it's a little disturbing because this has been around for at least 20 years. And so my question is, what needs to be done in order to bring some consistency or predictability among regulators in acceptance of these different approaches?
Anybody want to take that, Vikram?
Well, I mean, maybe I can start and and maybe Lisa can add to this. I think this, the way I look at this, so at least this is just my personal view. So I tend to have a greater belief with concentration response analyses. And that's intrinsically how I've been trained and what I have experienced. I have seen very few examples where if we have a clear understanding of the relationship and we're pretty comfortable with aspects of interpolation. Where I think things get a little tricky is when we are extrapolating. And I think that's where the large uncertainty resides. And there's a lot of reasons for that. From a regulatory perspective, I think we tend to be very afraid of precedents. And for some reason, if we do something for the first time, someone is going to come in and say, you did it there, why can't you do it here? Which I think is, a legalistic way of approaching these problems, not hugely a scientific way of approaching these problems. So my inherent sort of core is, I tend to believe these models. I think the way I look at it, it's companies are making remarkably large bets, decisions on moving these molecules forward, and they're doing it on very, very similar analyses. Coming back to, I think, why we are still struggling with this whole concept is, I think probably, I think the pharmacometric statistical community, I think, probably needs to sit and talk a little bit about this. Clearly, there are methodologies in the statistical domain that I think either pharmacometrics has not fully leveraged or utilized to the fullest extent, and I think we should be thinking about those very carefully and trying to see, what are the core aspects that lead to sort of this distrust of the data, which is basically what it is. I personally feel very comfortable of making dosing recommendations between QD, BID, interpolating, or even for subpopulations. I think we do that very routinely. Clinical pharmacologists do that routinely all the time. Sort of taking this next step to a regulatory decision where we can actually do this based on models, or even taking the word models from basic clinical pharmacology concepts, I think is the next big step. And I think Dr. Woodcock said this very well this morning. I don't think we have any option but to do it. I mean, because how many studies are we going to keep running? And how long is it all going to take? So I think there is a little bit of work that needs to be done from our end. The other aspect, I think, is we also have, from a regulatory agency perspective, some level of heterogeneity in which different divisions approach these systems. I think we've got to acknowledge this. And, if we, within the FDA, would actually look at it, we would say that, divisions such as, say, let's look at the cardiorenal division, which is, incredibly what I would say, for a lack of a word, embracing and sort of believers of how you use these clinical pharmacology principles. And you have other areas which are probably not as embracing. And that could be a variety of reasons. Some of it is probably where those medical officers have their core training from, what has been their experience. So I think there's a lot of contributing factors that actually are leading to this sort of variability in how we make these decisions. But, push comes to shove, I think we have examples where we've done it. We would like to see this happen very routinely. And I think we clearly would want to set the playing field to be very, very, level playing field, so everyone knows what the expectations are when they do these kinds of analyses. I think we'll probably get there.
Melissa, a brief comment to add to this. Can statistical consistency of statistical methods help?
So I made one comment at the end of my remarks too, but I think the value of the exposure response or concentration response modeling is not being questioned by if we talk about discipline-specific review statisticians. It's when and where it's applied. And so the later it's applied in the development program, the more nervous we get, because then you're in the confirmatory stage, and it's tough to confirm if you're just trying to fit a model. If that's all we've got, if we can't get enough responses because they're rare events or something hard to find, then that's all we've got. And so we need to just be honest about, the decisions we're making based on a model. Where I have trouble is when we then try to go back to the same data, pick off a subgroup and attach a p-value to it and say, clearly this subgroup in this range needs a dose adjustment, and I'm confirming it. You're not confirming it. We have a model, we estimated it, and you'd have to do another study to confirm it. So it's sort of what you say about it, I think, that is one problem. And I'll just reiterate my suggestion before. If we really do think that heavier people need more drug, or people with good kidneys need more drug, or whatever the algorithm is, if we really know this, then if we know it earlier, then let's study the dosing algorithm. We don't have to study fixed doses. I mean, maybe I'm breaking the law here and Bob will straighten me out. But, we want to know what dosing regimen makes patients better off, right? So I'd get away from the fixed doses and go ahead and study the dosing algorithm. It's all about personalized medicine, so personalize the dose. That's what I would do. Personal opinion, of course.
Try to get to two more comments, questions, if we can do them quickly, so please go ahead.
Alistair Breckenridge, United Kingdom and NDA partners. I'm giving the last talk of the day on perspectives on patient adherence. But many of you may not be here by that time. So the question I want to put to the panel is, how common is it to ensure patient drug adherence using reputable methods in clinical dose response studies? And if not, why not?
Not an easy question. Any takers for a quick response?
Well, I'll give it a whirl. I think it's a work in progress. I think we've seen a great awareness. That awareness is we've become much more aware of it. Here I get my words out. And we're looking at methodologies. You first starts e-diaries, things like that. But there's new tools coming. We've seen some work from Novartis and some of the things that they're publishing. I think many of the companies are thinking as well. Years ago we had a presentation that we spend all this time thinking about it. Do we need to do something different based on age or gender? But the biggest variability parameter is the fact that patients don't take their drug. So we've recognized that. Now I think we have tools much better to help understand that. It's a journey.
Yeah. Let's go back to Michael's comment about, I know we're focusing on early use of modeling to get better dosing information. I think Michael pointed out that there are lots of ways to potentially learn in the post-market setting, too, through collaborations with health systems and the like. So maybe another direction to go to. Bob?
Only that doing an analysis based on compliance is going to make everybody very nervous. Trying to encourage compliance during the study, that's fine. But, there's a long history of worrying about that. I just wanted to talk about when you can make use of concentration response data without worrying too much about it. It's worth noting we adjust for renal dysfunction all the time. We don't even think about it. We have no data. We just make the adjustment. The area that's brought a lot of thinking out about this has been the new anticoagulants like Apixaban and other drugs, where we have fabulous concentration response data and very steep curves for both bleeding and stroke. So it has a certain plausibility to it. I believe if you'd asked the group that looked at it, we would have approved a dose of Apixaban higher than had ever been studied. But we took it to an advisory committee. That made them very nervous. So what we did was we banned use of Apixaban in people with normal renal function because their concentration is too low. All of this thinking about all of this stuff made some components of our biostatistics group very, very nervous. But we thought we really understood it and had mountains of data. And of course, what we really want to see is what Liz is talking about. We want to see a comparison of a fixed dose with a modified dose. And so there's a lot of interesting stuff to talk about in all there, but it's, there are very few drugs where you get blood levels to choose the dose, but the novel anticoagulants, new anticoagulants, I'm supposed to call them, new anticoagulants seem like candidates for that. We've been doing it for warfarin for, a million years. Would it make sense to do that, and could you get people into the right place on a very bad disease with a very bad side effect? Anyway, I'll talk a little more about that.
All very interesting. Any final comments on this? I do want to, we will come back to this topic later on today.
No, this is a totally different topic, but I just wanted to talk about the Apixaban example that we had and the use of the utility endpoint. I'm a big fan of utility endpoints anyway because I like thinking about risk-benefit at the patient level. And, a patient with benefit and with risk but benefit is different than, thinking about benefit-risk at the population level. But this is one place where it seems to me that's a no-brainer, that putting the benefit and the risk together when you're trying to find the dose, why isn't everybody doing that? I just wanted to make that comment.
Great. Thanks very much. I'd like to thank our panel for an excellent discussion on a very important topic. We are going to come back and deal with more of this later, and because I want to make sure everybody is going to be here for Alistair Breckenridge's final presentation, I do want to promise you we will end on time. That said, because we are starting lunch a bit late, I don't want to rush people through that, and this is a good time for interactions with each other. We'll start 10 minutes later than scheduled, so at 1:55 rather than 1:45, and I promise we will end on time. Thank you all very much.
Got all these teed up.